There are over 300,000 species of plants, and there is huge variation in their appearance, form, or external structure. For example, some herbaceous plants like grasses and vegetables. Then we have woody plants, for example, vines which trail along the ground unless offered some type of support. We have shrubs which have multiple stems near the ground, and we have large trees with a single hard fibrous stem and a considerable height. Plant physiology deals with the study of functioning of plants, and functioning of plants is highly related to the structure of plants, what we call as the structural functional relationships. In this introductory lecture, I will introduce you to the basic structure of the plant and how the structure is related to different functions, like how plants photosynthesize, how they take up water and nutrients from soil, etc. Plant morphology or phytomorphology deals with the physical form and external structure of plants. Whereas plant anatomy deals with the study of internal structure of plants. Plant anatomy is mostly studied at the cellular or microscopic level. If we look at the structural organization of a plant consists of two organ systems, shoot system and root system. Shoot system is the aerial part or above ground part of the plant, whereas root system is the below ground part of plant. A shoot system consists of stem with leaves and branches, flowers and fruits. So this makes up shoot system. Whereas root system consists of roots and its associated structures. The primary function of stem is support and transport within the plant. The primary function of leaves is capturing light and converting it to chemical energy through the process of photosynthesis. Flowers and fruits are reproductive parts. Flowers attract the pollinators, whereas fruits are means of seed dispersal. The below ground part that consists of root system, the basic function of roots is the uptake of water and minerals from soil, the anchorage of plants into the soil, and sometimes they act as storage pods. The organ system is composed essentially of three types of tissues: ground tissues, vascular tissues, and dermal tissues. Whereas plants grow at meristems. Meristems are localized regions of active growth. Their purpose is cell division to form new cells. These new cells then differentiate to become specialized. Differentiation is the process of taking up a permanent shape, size, and function by a cell. Broadly, we can categorize plants into two types: monochords and dipoles. Monochords have one cotyledon and dipoles have two cotyledons. Cotyledons are the first leaves developed from the seed, or they are the embryonic leaves. The monochord plants are usually herbaceous. And dipoles can be herbaceous or woody. Herbaceous plants have a flexible stem, whereas woody plants have a hard fibrous stem, usually with a bark. Monochords have hypogeal mode of germination, whereas dipoles have epigeal mode of germination. Germination is the ability of a seed to produce a plant. More technically, seed germination is the emergence and development from the seed embryo of those essential structures which, for a kind of seed, indicate its ability to produce a normal plant under favorable conditions. Hypo means below ground, below, and epi means above. In hypogeal mode of germination, during the process of germination and afterwards, the seed remains below the soil surface. Whereas in epigeal mode of germination, the seed or cotyledons come above the soil surface. So monochords have hypogeal mode of germination, whereas dipoles have epigeal mode of germination. Monochords have parallel leaf venation, that is the arrangement of veins in leaves. So they have leaf veins parallel to the midrib, whereas dipoles have a net-like leaf venation, or technically reticulate leaf venation, like this. Monochords have a fibrous root system. That is a dense mass of thin hair-like roots, whereas dichords have a taproot system in which there is a main root and with minor or side roots. So this is usually a shallow root system, whereas the dichords have a taproot or deep root system. Some examples of monochords are wheat, rice, maize, and some examples of dichords are cotton, soybean, alfalfa, and many trees. Stopper. Mustard field, which is a dichord, and let's have a look at its structure. As it is a dichord plant, you see the net-like leaf venation, unlike monochords which have parallel veins. So this is net-like leaf venation, and it has a taproot system. You see there is a main root and then there are some side or minor roots. So this taproot system is usually deeper than the fibrous root system of monochords, and it helps to extract water and nutrients from relatively deeper layers of soils. So this below ground part is the root system, and this above ground part with stem, leaves, and here you see flowers is the shoot system. This is a rice plant which is a monochord, and you see its leaf venation. It has parallel leaf venation or parallel leaf veins, if you can see this. And this rice plant has dense mass of hair-like roots. This is called the fibrous root system, in which there is no main roots but a dense mass of roots, and usually they are shallow rooted or their depth is shallow. Plant tissues are made up of individual specialized cells, and they can be categorized into 4 types. That is meristematic tissues, dermal tissues, ground tissues, and vascular tissues. The meristematic tissues are made up of meristematic cells. Meristematic cells are thin-walled with a distinct nucleus, but they have no vacuole. The basic purpose of meristematic tissues is cell division, to increase the number of cells. These cells then differentiate and become specialized. Differentiation is the process of taking up a permanent shape, size, and function. For example, cells, the meristematic cells differentiate and become epidermis, and which may form stem and leaves. They do not need a vacuole because these cells are growing actively and they do not need storage or support, so they lack a vacuole. So meristematic tissues are regions, localized regions of active growth. They keep on dividing and dividing and dividing and increasing the number of cells which can become differentiated and specialized. So the basic purpose of meristematic cells is cell division and growth, which can be primary growth or horizontal growth or secondary growth. Primary growth is the vertical growth. Plants increase in length. This is primary growth. Whereas secondary growth is the horizontal growth. That is the increase in stem girth or increase in the diameter of trunk. That is horizontal growth or secondary growth. So the basic purpose of meristematic tissues is primary or secondary growth, and they are localized. It means they are located in specific parts of the plants, and based on their location there are 4 different types of meristematic tissues. First is apical meristem. Apical meristems are located at the apex or at the tip of plant, tip of root or tip of shoot, what we call as shoot apical meristem, and tip of the root what we call as root apical meristem. So the root apical meristem is further protected by a root cap. The basic purpose of apical meristem is vertical growth. So increase in plant height, both upward and downward. So they help the plant shoot up or shoot down. Second type is intercalating meristems. Intercalating meristems are located at the base of nodes and internodes. Nodes are enlarged portions of stem where leaves are attached and where vegetative or floral buds initiate. And the distance between 2 nodes is called as internode. Intercalating meristems are located at the base of these nodes and internodes, and they are responsible for regrowth. For example, regrowth of grasses. When grasses are eaten by herbivores or animals, or when we cut the long grass, we see a rapid regrowth. So this is due to the presence of these intercalating meristems, and this is also a plant defense mechanism. So they play their role in plant defense by giving the plant's ability to regrow when cut intentionally by landscapers or when they are attacked by herbivores. They are also found in certain plants like bamboo, and they are responsible for rapid growth of the internodes. So intercalating meristems are found at the base of nodes and internodes, and they are responsible for regrowth of plants or rapid growth of plants. Axillary meristems. Nodes are enlarged portion of the stem to which leaves are attached and where vegetative or floral buds initiate. The angle between these leaves and stem is called as axil. Nodes are the points where the buds initiate. This is a bud, axillary bud or lateral buds. These buds become branches or flowers. For example, in soybean, these buds develop into flowers which are, after fertilization, become pods containing seed. So axillary meristems are responsible for the development of buds that develop into branches and flowers. This type of meristematic tissues is secondary meristem. Secondary meristems are responsible for lateral growth, so they are also called as lateral meristems, and they are found in lateral parts of the plant. This is the secondary meristems. Secondary meristems have two types: cork cambium and vascular cambium. Cork cambium is found in woody plants where it produces tissues that replace epidermis. And vascular cambium is found in stems and roots of dicot plants and is responsible for increase in stem diameter or horizontal growth. The activity of vascular cambium can also be observed in the annual rings formed in tree trunks. From these annual rings, we can estimate the age of a tree. These annual rings are formed one cycle of hardwood alternating with this sapwood or light colored wood. So the formation of these annual rings is due to the activity of this vascular cambium. And secondary meristems or lateral meristems are responsible for lateral growth of plant. So meristematic tissues consists of actively dividing meristematic cells, which are thin-walled, lacquer vacuole, but they have a distinct nucleus, which when divide, they keep on dividing, and they are responsible for vertical or horizontal growth of plants, and these are localized regions of active growth. They are located in specific parts of plants, and based on their location they can be called as apical meristems, intercalating meristem, axillary meristems, or secondary meristems. Apical meristems are found at tip of the root and shoots, and a phenomena related to apical meristems is called as Epical Dominance, that the terminal bud prevents the or inhibits the growth of lateral buds, and plants keep on growing in vertical direction. And when we remove this bud, sometime on purpose, for example, if we want to curtail the height of a plant for ornamental purpose, or we want to induce the reproductive growth, then we remove this bud manually or through certain chemicals, and then the growth of lateral buds, and we have intercalating meristems at the base of nodes and internodes. They are responsible for regrowth or for rapid growth. The axillary meristems are found in leaf axils, where they are responsible for the development of vegetative or floral buds, and secondary or lateral meristems are responsible for lateral growth. Discussing the meristematic tissues, let's now have a look at the dermal tissues. Dermal tissue is the outermost protective covering of entire plant body. That is roots, stems, leaves, fruits, etc. So this dermal tissue is comparable to our skin, that is in direct contact with its environment, and its role is protective. But the main difference between our skin and the dermal tissues is that usually it is single-celled, very thin, compared to our skin that is multicellular. But this can also be multicellular. Usually it is one cell thick, consisting of elongated dermal cells, and its role is protective. So what does it protect plant from? It protects plants against the loss of water, because water is a precious commodity. Water, although it is the most abundant, yet it is the most scarce commodity, the most limited commodity for crop production. So plants try to maintain their water status or water level within the plant body, and dermal tissues help prevent the loss of water. They also protect plants against pathogens or disease causing organisms like bacteria, fungi, nematodes, etc. And in terms of loss of water, they are also responsible for the protection against loss of water by, for example, a horticulture commodity like fruits after the harvest. So how fast a horticultural commodity loses water by transpiration depends on the type and thickness of these dermal tissues. So dermal tissues protects against loss of water, against pathogens, and against the penetration of chemicals. So dermal tissues, for these reasons, is also called as protective tissue, that is in direct contact with its physical environment. The dermal tissue consists of different elements like epidermis, cuticle, stomata, lenticles, and trichomes. If we look at cross section of a leaf and draw it here, we see a single cell layer of elongated cells. This is called as the epidermis or upper epidermis. Then we have mesophyll cells, which have their role in photosynthesis, and here we have vascular tissues, then lower epidermis, the epidermal cells, which are elongated cells, they secrete a fatty substance called as cutin. This cutin helps the formation of another protective layer above the epidermis, that is called as cuticle or cuticular membrane. This cuticle prevents the loss of water and prevents the entry of pathogens. So it keeps water in and microbes out. Water within the plant and microbes or pathogens outside the plant. And in lower epidermis, this is lower epidermis, you see small openings here, which are called as stomata. These small openings or stomata or stomats are responsible for exchange of gases, carbon dioxide and oxygen, as well as exchange of water vapors. Plants lose water in the process of transpiration, and 90% of transpiration is done through these stomatal openings or stomata. So epidermis is the outermost covering consisting of elongated cells. These cells secrete a fatty substance cutin, which form another protective covering, cuticular membrane or cuticle. And how thick this cuticle is determines how fast a plant loses water. And this epidermis is usually one cell thick, but in cases of water short environments or water limited conditions, this can be multicellular thing. And this has openings called stomata. These are the small openings which are highly regulated by plant. These stomata can be opened or can be closed depending on the soil environment. For example, in the morning when the temperature is low and when there is abundant water available in the root zone, the stomata opens and plants rapidly lose water by transpiration, and at the same time these stomatal openings allow the entry of carbon dioxide into the mesophyll cells, which is then fixed through the process of photosynthesis to produce sugars. While at daytime, at noon, when there is a high temperature or there is a shortage of water in the root zone or root environment, then these stomats close to prevent the loss of water by the plants. So that's how the dermal tissues protect against the loss of water as well as against the temperature stress, making plant able to survive under adverse conditions of heat stress or drought stress. Then we have trichomes, which are usually single-celled, hair-like structures found on stems or fruits, for example in peach or in kiwi fruit. They are also responsible for preventing the loss of water, or sometimes they offer resistance to attack by pathogens. Lenticels are also small openings in stems or petals or flowers. These are also small openings like stomata, but the main difference between stomata and lenticels is that the stomates or stomata can be regulated, they can be opened or closed by plants, whereas lenticels always remain open. Like stomata, they are also responsible for exchange of gases like carbon dioxide, oxygen, and for the loss of water. So the dermal tissues, to summarize, are the protective tissues. They are comparable to our skin. They consist of different elements like epidermis, stomata, lenticels, and trichomes, and they protect the plant against loss of water, against pathogens, against temperature stress, and against penetration of chemicals. Third category of tissues is ground tissues. Ground tissues are also called as simple permanent tissues. They are tissues other than the epidermal or vascular tissues, such as pith and cortex. For example, in dicot stems, the ground tissue is divided into two regions, cortex and pith. This is a dicot stem with the outermost covering or epidermis, and this is the vascular cambium that we discussed in lateral meristems. And here there is primary xylem and flowing or vascular tissues. And outside this vascular bundle is the region called as cortex. Outside or around this vascular bundle or vascular tissues. This region is called as cortex, and this region towards the inner side of the stem, this is called as pith. These cortex and piths are composed of parenchyma type of cells. These ground tissues are further categorized into three types, or they are made up of three types of cells. Parenchyma cells or parenchyma tissues. Then we have collenchyma tissues, and the third type is Scleritimer tissues. This parenchyma tissue makes the bulk of the plant. If we talk about the main functions of these ground tissues, these are very important tissues that play many functions in plants. For example, they make up the edible part of fruits and vegetables. Since they are bulk of the plant, so next time if you are eating a fruit, technically you may be eating parenchyma cells or parenchyma tissues. They are responsible for storage of energy, water and nutrients. They are responsible for important plant processes like photosynthesis, respiration and conversion of glucose to starch. They also impart flexibility to the plant. As we discussed that herbaceous plants are those plants which have a flexible stem. So this flexibility is given by ground tissues. And in addition to giving flexibility to the stem of herbaceous plants or leaves or petals, they also give toughness or rigidity to certain parts like husk of coconut or peanut. So they give flexibility as well as rigidity to the plant. So ground tissues are responsible for storage, for important metabolic processes, for giving flexibility as well as rigidity to the plant. And they are also responsible for another important process, that is wound healing. So when a plant is injured by an animal or in case of mechanical injury, then it can heal itself, certain cells, and that is through these ground tissues. So these are very important tissues that have many functions or many roles to play in plants. As I told you that ground tissue consists of further three types of simple tissues or three types of cells. So now we look into more detail of these three types of cells: parenchyma cells, collenchyma cells and scleritimer cells. Parenchyma cells, when they are first made, they are spherical in shape, and then they get packed, tightly packed, side by side. They have many important roles to play in plants, and they make up the bulk of the plant. So they are found in all plant organs. For example, in leaves, they are called as Parenchyma cells. This is a specialized form of Parenchyma cells which contain chlorophyll, and they are responsible for photosynthesis. Another type of Parenchyma cells is Parenchyma cells, which have large intercellular spaces. This is the intercellular spaces. And this is important, for example, in rice, which mostly grows in transplanted and flooded conditions, and there are certain parts of the plants that are submerged in water, and plants need oxygen for respiration. The supply of oxygen to those submerged parts of rice plants are made possible by these Parenchyma cells. So that is how rice plants can survive under flooded or submerged conditions, where most of other plants fail. And this also gives buoyancy to certain plants. For example, water leveling buoyancy is the property of floating on the surface of a liquid or in a fluid. So these Parenchyma cells also give this property to certain plants like water lily. Other roles of Parenchyma cells are storage of energy, water and nutrients. They make up bulk of the plants, and they are found in edible parts, and they are also responsible for wound healing in plants. Second type of tissues is Parenchyma tissues, which are made up of Parenchyma cells. Parenchyma cells are similar to Parenchyma cells, but their walls are thicker and uneven, and their cells are more elongated than the Parenchyma cells. This thickness of cell walls is due to the presence of cellulose and pectin, and this thickness gives strength or toughness to the plant. But these cell walls or secondary cell walls are uneven. This unevenness gives, in addition to strength or toughness, this also gives pliability to the plant. This makes them pliable or flexible. So they are found in plant parts like young stems, like in leaves, veins, or in flowers, and gives flexibility to the plant so that they can move around. So Parenchyma cells are similar to Parenchyma cells, but their cell walls are thickened, uneven, and their cells are elongated. In addition to giving strength to the plant parts, they also give pliability or flexibility due to which stem of herbaceous plants or leaf petals or flowers can move. Parenchyma and Parenchyma cells have they are living cells, or they are living at maturity. The third type of cells, which is called as Scleritimer cells, they are mostly dead at maturity. and their main purpose is structural support. These cells have thickened secondary cell walls which are highly lignified. So these cells contain cellulose and lignin. Lignin is an organic substance that gives super toughness to the plant, and their thick secondary walls, they are thickened by the presence of this lignin. They are also called as lignified. So in addition to the structural support, they are also responsible for transport of water and nutrients throughout the plant body. These Scleritimer cells are found in hard parts of the plant, such as husk of a coconut, covering of seeds such as nuts, in leaf veins, as a stone of drupe fruits such as cherries and plums, etc. So they are found in harder parts of the plant. They impart super toughness or rigidity to the plants. Scleritimer cells are further categorized as non-conducting and conducting cells. Non-conducting cells are sclerites and fibers. Sclerites have a variety of shapes. They can be oval or round or cylindrical, whereas fibers are elongated cells. Sclerites are distributed throughout the plant, whereas fibers are usually associated with vascular tissues, xylem and fluid. The conducting cells, are also called as tracheic elements, consist of tracheids and vessels. If we talk about tracheids and vessels, or the tracheic elements, these tracheids have a smaller diameter, whereas vessels have a broader diameter. Tracheids have no perforations, whereas vessels have perforations all over. Tracheids are shorter in length, whereas vessels are longer in length. Tracheids are found in all vascular plants, whereas vessels are found in angiosperms, or the plants that have flowers and produce seeds enclosed within a carpel. So Scleritimer cells are mostly dead at maturity, and they give structural support to the plant, as well as they are responsible for transport of water and mineral throughout the plant. So ground tissues are tissues other than the epidermal or vascular tissues. They make up bulk of the plant. They have many important functions to the plant. They are the site of important metabolic functions like photosynthesis, respiration, etc. They are responsible for giving strength to the plant, as well as giving flexibility, and they make up bulk of the plant and are further comprised of three types of simple tissues: parenchyma tissues, collenchyma tissues, and sclerenchyma tissues. tissues. Last type of tissues that we are going to discuss is vascular tissues. Vascular tissues are xylem and phloem. Xylem and phloem, they are also called as vascular bundle or conducting tissues. So they form a tunnel-like system, or they make up the plumbing system of the plant, through which water, minerals, hormones, and food can move around and within the plant. In dicot stem, the vascular bundle is present in the form of a ring. So this is primary xylem and this is primary phloem. So they are phloem. So they are present in the form of a ring. Whereas in monochord stem, the vascular bundle is scattered throughout the plant body like this. So they are not arranged nicely in monochord stems. So this is how we can differentiate the monochords and dichords based on vascular bundle. The main difference between xylem and phloem is that xylem transports water and minerals in one direction. So it takes up water and minerals from soil. So it moves water and minerals from root system to shoot system. So this is unidirectional movement or one-way movement. Whereas phloem distributes the products of photosynthesis, mainly sucrose or some other organic substances, in two ways, both upward and downward. So it can move the products of photosynthesis from leaves to upper parts of the plant as well as down to roots. The xylem that transports water and minerals from root system to shoot system is made up of 4 types of cells. Xylem Parenchyma cells, Xylem Fibers, tracheids and vessels. We have already discussed these parenchyma, fibers, tracheids and vessels in simple permanent tissues or ground tissues. So xylem is made up of different types of cells. So that is why the vascular tissues or xylem and phloem are also called as complex permanent tissues. So this complex permanent tissue xylem is made up of 4 types of cells: xylem parenchyma cells, xylem fibers, xylem tracheids and xylem vessels. Xylem parenchyma cells, their main purpose is storage of food and sideways conduction of water. When water is moving from roots to stem, then sometimes plant also needs to move water sideways for even distribution of water throughout the plant, and that is made possible by these parenchyma cells, which can store water or conduct water and store food. The xylem fibers, as we discussed in Scleritimer cells, they give structural support to the xylem. Trachees and vessels are dead at maturity, and they actually make the conducting system of the xylem, or they actually transport the water and minerals from roots to other parts of the plant, or from root system to shoot system. And they are super tough due to the lignification of secondary cell walls. So a xylem is responsible for unidirectional movement of water and minerals, and this is a complex permanent tissue which is made up of 4 different cells: parenchyma cells, fiber cells, trachees and vessels. Second type of vascular tissues is phloem. That is a Greek word which literally means bark. And the purpose of phloem is transportation of the products of photosynthesis, or what we call as photosynthetes, like sucrose, from leaves to other parts of the plants. Phloem is also a complex permanent tissue that consists of 4 types of cells: sieve elements, companion cells, parenchyma cells, and fibers. Scleritimer or phloem fibers. Sieve elements or sieve plate, this is the long tubular structure with perforations at its end that helps in transport of food in 2 directions. These companion cells are specialized parenchyma cells that help maintaining the pressure gradient and support these sieve elements or sieve plates. The phloem parenchyma cells are responsible for storage of food in the form of starch or proteins or fats or tannins in some cases. And phloem fibers help in structural support of the plant. So in this way, the phloem transports the products of photosynthesis and distributes it throughout the plant body where they are needed. Just to summarize what we have discussed so far, the plant structure is closely linked to its functions. For example, if we have 3 varieties of a plant, and variety V1 has horizontal leaves, V2 have erect leaves, and V3 has intermediate type of leaves which are neither horizontal nor erect like this. This is variety 1, this is variety 2, this is variety 3. What do we expect in terms of light interception? Which plant will intercept more light? Then the answer is this intermediate type of plant, or V3, will intercept more light, will absorb more light, and probably will do more photosynthesis because of more absorption of light. In case of V1, the upper leaves will intercept maximum light, but they will shade the lower leaves, so there will be less interception of light. Whereas these leaves have an angle, they are so erect that they will have minimum light interception, and this intermediate leaf architecture type variety will have maximum light interception and so maximum photosynthesis. So in this way, plant structure is related to its function, and that is important when we develop the crop management practices. For example, in sesame we have two different varieties. One is single stem variety that is called as TH6, and one is branch variety that is called as what we call as TS5. So when we are planting these different varieties, this variety needs more spacing, more row to row spacing, as well as more plant to plant spacing compared to this variety that is single stem. So while planting, the seed rate will also be lower for TS5, as well as the spacing must be wider. So we have more row to row and more plant to plant distance in case of TS5. So that is how plant structure is related to its functions, and keeping in mind these structure function relationships, we develop the crop management practices. If we talk about structural organization of plants, we can discuss the two main organ systems: the below ground organ system, that is called as root system, and the above ground organ system, that is shoot system. Shoot system consists of a stem with leaves and may have fruits and flowers. The basic function of stem is support and transport. Basic function of leaves is capturing light energy and converting it into photosynthesis or chemical energy. The basic function of fruits and flowers is dispersal and survival of plant, and the basic function of roots is anchorage into the soil, transport of water and minerals, and in some cases storage. Broadly, we can categorize the plants into two categories: monochords and dipoles, based on the number of cotyledons, their leaf venation, and their mode of germination, and their type of root system. Plant tissues are composed of specialized individual cells, and we can categorize plant tissues into four types. Plant meristems, where actually plants grow or divide to form new cells and new tissues, which become differentiated to become ground tissue or vascular tissue. And then we discuss different types of tissues, like based on location of meristematic tissues. There are four types: apical meristems, axillary meristems, intercalating meristems, and secondary meristems. We also discuss the dermal tissues and its different elements like epidermis, stomata, lenticles, trichomes, and cuticle. We discuss three types of ground tissues, which are parenchyma, collenchyma, and sclerenchyma tissues. And we discuss the structure and function of vascular tissues or xylem and phloem. So that is how the structure of plants is related to different functions.